Thank you, Navya, for the introduction. Hello, everyone. It's great to have you all here with us. In this session, we will discuss the unique Omix Logic project-based training program, which is Introduction to Big Data Bioinformatics, as you can see on the screen. This is a special fall program to make bioinformatics accessible to students and researchers to learn about cutting-edge omics research, bioinformatics, and computational biology. And we will learn to analyze complex data sets to answer important research questions. So in the next few slides, we will learn more about the topics, resources, and the outcomes associated with this 1 month training program. And I hope that I'm audible to all of you. And
Yes,
if you
Dr.
have any,
You're
thank
audible.
you, Dr. Prit. And if you have any questions throughout the presentation, feel free to put it in the chat. And I hope that once that, as Navya have showed you all, you have logged into the Learn Omix Logic portal, which we will be using throughout the presentation. So this research-based training is scheduled to start on 24th September 2021, and with a live orientation session that we will be going through in much more detail in a while. The advantage of learning something new is that we all start from the beginning, right? So with this incredible growth in big data, big data has turned out to be a remarkable career choice for many today. Industries from various domains offer different positions for big data professionals. That includes biology and healthcare too. So from a data perspective, what makes biology unique from other realms is its sheer size. For example, typical genome of a microbe, which might have 40,000 genes, if we assume that there are 1,000 distinct variations or variants of each gene, that would require us to conduct 1 billion tests. So just imagine the amount of information that could be generated from this sort of analysis. So biology in reality is rapidly acquiring the characteristics of data science, and billions of data points on genes, proteins, and other molecules are compiled in large files and are systematically studied. So analyzing this data will lead to more knowledge and understanding about living organisms, including human health, biotechnology, crops, and livestock. So it includes a lot of application in that context. So this course will give an overview of bioinformatics for students, biologists, clinicians. So the influx of data associated with methodologies and data-driven research, making this bioinformatics for life science as one of the prominent and emerging technology that one could start learning about. So it would involve learning about computational methods, and it will involve learning about the biology and the biological research associated with it. To give you an overview, I would wanted to let you given a basic understanding of NGS-based techniques and its translation application, because it's one of the key research areas in today's world. So NGS is beginning to provide these powerful insights into genetic mutations, molecular networks, underlying diseases for a broad set of genes. So let me ask you a simple question. What is NGS techniques? And please feel free to write it in the chat box, your answers. So I'm just asking, like, what is NGS? Can you tell us? Sequencing technology. Vanishka, thank you for answering. Zara says Illumina. Okay, Illumina is a sequencing company. Nupur is saying next generation sequencing technology. All right. Yeah, next generation sequencing. So next generation sequencing, or NGS, that's the full form for next generation sequencing or high throughput sequencing, is a massively parallel sequencing technology that offers ultra high throughput scalability and speed. So the technology is used to determine the order of nucleotides in entire genomes or targeted regions of DNA or RNA. I hope that clarifies doubts if you have any. So the next question I would wanted to ask you all is, what is the role of bioinformatics in NGS? Why we use bioinformatics for NGS? Lisa, yeah, tools to investigate genomic questions. That's a good answer. Anyone else? Data interpretation. George, thank you. So we are discussing here about a program which is on big data bioinformatics. And we are talking about big data, and data becomes big and big in size when the data have few characteristics. So we define big data by few characteristics, and it's good to see you all answering different aspects, such as Nupur here is saying to study different data sets of genes. Yeah, in a way, yes. So next generation sequencing technologies offer high throughput, rapid and accurate methods for determining the precise order of nucleotides in a given within a DNA or RNA molecule, right? That's what I said. So the combined power of NGS and bioinformatics kind of play vital role for different applications. It plays vital role for diagnostics that has been proven. It plays role in medical treatment and epidemiological research. NGS technologies are currently used for whole genome sequencing that many of you have heard of, and it helps in investigation of genome diversity, as Lisa said. And also there are other applications which are very new, are getting much more popular because of its vast integration ability or data interpretation ability, as George said. So there are different fields such as metagenomics, epigenetics, then discovering non-coding RNAs, protein binding sites, and then the gene expression profiling, which is the RNA-seq. So what we did learn a little bit is that why we should learn and understand and analyze high throughput data. What is the need and why we should be doing it, and why should we be learning about it? We need to do that because big data now has application in almost all the technologies, right? And it's equally important to learn this aspect in terms of biology, in terms of life sciences. So now, like, as we had an overview of what we are doing, and I wanted to tell you just about like the OmicsLogic program or this training. So OmicsLogic is a program that embeds data-driven concepts into biological projects. So it's a project-driven training. So we will be all the time describing examples taken from publications, examples taken from NCBI datasets. So spanning the student learning journey from an observer to participate in research. So when we talk about training, we usually think about delivery lectures and sessions, and sometimes we also have opportunity to do some hands-on. Here is the opportunity to do research, and that's why it is a project research-based training. So this journey begins with identifying a interesting biological research question, or a question that can be answered with data. Because in bioinformatics, we kind of look for data-driven approaches, and not in bioinformatics. If we talk about data science, machine learning, those are all data-driven approaches, right? So the journey begins with identifying question or a research problem, and then we collect data, we organize data, and we understand by analyzing that and drawing conclusion from it, right? And this journey could start from from the login on the Learn portal. I mean, it's as simple as that. So all of you, if you haven't done so far, please make sure that you created login, because with so much data available on biological topics and questions, learning about this exciting field and application is a pathway to new research and opportunities. So please, I'll make sure that you have a login on the Learn Omics Logic. So this Omics Logic curriculum, actually, it follows, as I said, project-based, student-centric learning model, where our focus is to simplify complex bioinformatics methods and use these methods efficiently and find meaningful information to tackle the unsolved problems in biology and healthcare. And I'm telling you, it's pretty exciting. So this online program is actually, as Navya said, is designed for everyone, including students and participants who do not have a background in bioinformatics and are biologists or clinicians who wants to learn about big data, high throughput data, engines, data analysis, processing sections. We will be covering in this program genomics, as I said, transcriptomics, and biomedical data science and machine learning. So looking at biology, the big data in biology, and being able to gain insight. So it has several applications, from research to discovery and to integration of system biology. So as a result of this, the participant will have an opportunity to become more proficient in the analysis, interpretation of various types of omics data, read through publications, understand publications, and the different type of research that is being conducted. So it includes statistics and machine learning. So let's begin with the review of topics. And again, I wanted to insist that if you have any questions, please feel free to put it in the chat box. So as I was saying, that let's review the topics that we will discuss in depth. So during this 1 month training, we will cover important topics that are used by the biomedical and biotechnology industry, the 21st century problems. So that would include introduction to big data in biology. Then we will do introduction to genomics, mapping and sequence visualization, big data, obviously. And then introduction to transcriptomics, gene expression data analysis. Then we will learn about differential gene expression. Then we will learn about biomedical data science that would involve learning Python and R for data analysis, not like not as a programming language, but as a tool. And then the application of this into a real machine learning project, NGS-based project. So these are some of the key topics that we will be covered. And let me now go into much more detail so that you get a clear understanding of how this program will be and what are the resources that will be and how to utilize that with with best approaches and best ways. So in the first session and the first week of this training, starting 24th September 2021, we will have introduction and commencement of the program. Then we will have introduction of the faculty, mentors, and trainers. Then we will demonstrate you the Slack channel. So there's a Slack channel associated with each of these programs where we have all the participants together so that we can communicate about the resources, the meeting links, and all of that, like any sort of questions, technical questions that you have. And we will have also mentors added to the Slack channel so that they can answer to your questions. And then there is a program page. So the program page where you get all the schedule, all the other resources, we will showcase you that in a while. So in this session, we will be taking an overview of the resources, and it's kind of an orientation session where you will be getting the passwords and the access to the platforms such as the Learn platform and the Bioinfo platform. Then there will be a Q&A discussion where you can ask your questions similar to this session. And what are the expectations, schedule, and review of the training program, description of the structure of the course, and deadlines. So that is something that we will be discussing on the first session, making everyone having all the resources, connecting for collaborative work. So we have been discussing about the platforms. So one of the platforms that we will be utilizing, and it is something of a core of what we do, is utilize this Bioinfo platform along with open source resources. So the Bioinfo platform was developed at the Tauber Bioinformatics Research Center by computer scientists and mathematicians passionate about big data integration. So to make the power of advanced algorithms accessible, the system allows for a user-friendly and logical data analysis interface that can be used by anyone. So with biological discovery in your mind and a focus on advanced methods like mathematical modeling, machine learning, and efficient computational tools, this platform can actually take you further in a very short amount of time. So there are several sections that we will be utilizing in this program itself, which includes the transcriptomics gene expression data analysis section, the data mining section on machine learning and utilizing different network analysis, and then you will be able to see and manage your own pipelines and see what kind of analysis that you have run. So the resources and the data, tells us a clear story. So that as a scientist, as a researcher, as a student, anyone is capable of becoming scientist now and has the opportunity to do the research by. So this growth in data has allowed researchers to sequence individual genomes and match combinations of mutations with specific diseases. I'll give you an example of cancer. As cancer is inherently a disease of the genome, it is not surprising to see NGS technology already becoming applied to cancer research with promises of greater understanding of carcinogenesis, right? So while the task of deciphering the cancer genome code is still ongoing, we are already beginning to see the application of genetic-based testing or diagnostics or biomarkers in the area of cancer. So this program or this course is designed to highlight the role of big omics data that is shaping modern biomedical innovation, where molecular data is used for precision diagnostics, big data integration, and machine learning is transforming personalization of treatment selection, and innovative algorithms allow for unprecedented capabilities for drug discovery, designing even of large molecules and drug repurposing. This week, this is the first week that we are describing. In this week, after learning about the introduction to bioinformatics and bites in the molecule, we will learn about the topics of genomics, topics in genomics. So what we will cover is genomic variation, which will be a detailed understanding, the types of different sequencing methods, targeted sequencing, whole genome sequencing, whole exome sequencing, and then we will understand about the logical steps for genomic data analysis and what are the associated algorithms. Then we will look analyze the results by visualizing using the integrated or integrative genomic viewer. So genomics, as I said, is an essential subfield of bioinformatics and a major force in expanding human knowledge of genetically associated or associations with diseases and other traits. So it is an interdisciplinary field which includes the development of methods for DNA sequencing as well as for big data analysis of genomic sequences, right? So there are massive databases, and as I was telling you, that where the opportunity lies, and that opportunity lies in these massive databases of biological sequence data, such as GenBank, EMBL, SRA, that allows for data-driven research and knowledge discovery. So once the sequence or the raw data is available, it is required, or you need to process it to get and interpret biological insights, right? So we will be using defined logical steps that are applied to process the genomic data associated with various algorithms such as Bowtie 2, or here this is an example of Strelka. And to explore the genomic data, we would then visualize it with integrative genomic viewer to get a detailed view of the single nucleotide polymorphism, or the SNP. So in this first week, we will learn about various algorithms that are associated with genomic data analysis on the Bioinfo platform. And during this week, the second important aspect, as I was telling you, the participants will have the opportunity to complete the online courses ahead of the session. So you will have the access once you register for the course, and you will have access to all of these courses. So you can prepare, like take these courses and prepare ahead. So to ask your questions and which would be more meaningful, we kind of recommend to take the courses ahead. And if not, if you have missed a session, for example, then you will have an opportunity to cover all the important topics that are associated with the week learning using this resource. There's a link associated with it. So you will get an understanding of genetic associations provided by genomic research that has a great value in medicine, even in agriculture, even in ecology and biotechnology and many other industries. So as I was telling you, you will find out about different topics relevant to genomics while browsing through the courses. So this is course 3 on Learn Omics Logic portal. So if you have already created login, you can check out the resource and you will check out the online courses which will be available for you to take. So in the next week, after genomics, we will start learning about the important gene expression data, as I was telling you before. So in this, we will learn about the analysis logic, which is, I mean, from raw reads to table of expression, how we do that, and what are the common sources of unwanted technical variations? How do we, how do we remove those unwanted technical variations? What are the pre-processing steps? How we can filter and clean the table of expression? How we can load processing data for analysis? So we will learn about next generation sequencing, transcriptomics to understand the variation, and we will do practical. So we will do hands-on training on data sets. So today, I mean, if you are all available till the end, we will have a short demo session working with breast cancer subtypes and NGS sequencing data. So this project that we are talking about, it uses data from 2016 study by Bradford, James et al., where they did a whole transcriptome profiling of patient-derived xenografts models. So as a tool to identify both tumor and stromatal specific biomarkers. So the PDX, like it's a short form for patient-derived xenografts. What are they? I mean, you must have this question if you are not sure about what is PDX. I just said that it's a cancer subtype, and so what is PDX? So PDX or patient-derived xenografts are models for cancer in which human tumor samples are studied in a mouse environment. So in the study, they distinguished tumor and stromatal compartments by associating the stromatal compartments with murine mouse, which is mouse transcriptomic profiles. So utilizing the transcriptomic sections on the platform, we will be learning about analysis logic, as I said, from raw reads to table of expression, pre-processing steps, filtering and cleaning the table of expression. So this analytical platform, the Bioinfo platform, will provide hands-on experience for anyone to learn and apply data sets. And the sections are color-coded with a logical map. So explanation. So you'll find the explanation of methods, detailed input and output formats, and reproducible workflows when you go through and browse and go through this courses and training. So in this week, obviously, as we have it for genomics, we will be working on the transcriptomics section and learn on and use the Learn Omics Logic, where you will have the opportunity to go through the courses that are put together and covers important topics relevant to RNA sequencing, gene expression data analysis, and application. In the next session in that week only, where we are going through RNA-seq and we were like learning about RNA-seq and its application, we will have an opportunity to now dig deeper and understand the application in research, which is introduction to differential gene expression. We will learn about techniques or algorithms such as the seq-HR. We will learn about visualization or how to visualize this data by plotting volcano plots or heat maps. And then we will be learning about regression and factor regression analysis. That is a machine learning technique to understand about the factors and different types of conditions. So this will give you an idea of how transcriptomics is applied and how it's so much applied to biomedical research and different kind of research and its how can you do it, like with proper data sets that you can, work on. So during this advanced analysis of RNA-seq, as I was saying, data sets, we will be using two different data sets. One that you have already learned about, which is the patient xenograph one. And then we will be using a second data set that will help us understand more about the application of machine learning on gene expression data, how different types of data can be integrated to get an outcome or get an holistic understanding. So in theory, we will learn about in this during this entire week, like we will be learning about basic statistical techniques, tests such as student t-test. We will be able to perform statistical differences between two groups to learn about the key features. So I think it will be much more clear during the session because we have a dedicated session described for this, and obviously the online resources. So everything is also detailedly described. So to understand advanced visualization, interpretation of output, and then do the practical hands-on training on differential gene expression using the HR sequencing, DSEC, and such tools, you should try practicing on the transcriptomics section for the courses also on the Bioinfo platform, which we'll be covering in the session. So once we have practiced regression and other statistical techniques, we will progress to more advanced techniques. Well, these are the techniques that are changing the way data is being analyzed. And yes, I'm talking about machine learning, deep learning, and artificial intelligence. So during this week, we'll start learning about data mining and classification techniques. In this session, we will cover introduction to machine learning, types of machine learning methods, overview of supervised machine learning methods, which will be helpful for us in finding patterns from multidimensional omics data. We will be able to find similarities in the data, and to use that, we will use algorithms such as principal component analysis and clustering, such as hierarchical and k-mean clustering. So giving you an example, I mean, finding relationship in data is pretty important. Let's say we collected data on, for example, here we have data on eye color, gender, age, height, and weight of people. These are all clinically important data, right? We hypothesize that some variables here are related and some are not. We also think that there are exceptions to the rule. The age and height or weight are related, but not always, right? So relationships observed in data can show which variables are related, which variables are not related. For example, weight and height seems to be logically related. Are they in the data set? If we plot a trend line here, like you can see on the screen, if we plot a trend line, we will find that the trend line goes up. The higher the weight, the higher the height, weight also increases. But the dots are not too close to the line. Some dots seems to be far away from it, right? Is there a way to quantify this relationship that we found? So in this session, in this machine learning session, we will be understanding of the analytical methods for processing, visualization, and analysis of complex biomedical data. We will be learning about the terminologies that are very common and very important for machine learning and artificial intelligence in biomedical discovery. And we will be utilizing the Bioinfo platform, where we have the data mining section to do the statistical analysis and apply ML algorithms. So you will find this all information in course number 7, obviously associated with the session. And once we have got an understanding of how this application is, how we can use different types of machine learning and do data mining, next is to do predictive modeling or applying supervised machine learning methods. So in this session, the last session, the 6th session, we will be learning about decision trees, random forest, support vector machine, and then project examples. So in the project examples, we will also take you through what would be the next step. Once you have done this learning, once you have done all of this session, what can you do next? You can work on your own project, where we have the research fellowship program to support development of projects. And we have a lot of project examples that we will be that you will be having access to develop a specialization. So if you're interested in infectious diseases, whether you're interested in cancer, whether you're interested in malaria, or you have any other disease in your or any other problem in your for you to do research. So we have already a compilation of all of this, and as I promised earlier, I will be taking you to the way we can do, like, you can access all of these resources, register for the program. But before that, I would love to invite Dr. Harpreet to give you a short demonstration of one of the projects